My name is Maxim Kuleshov. I am a bioinformatician at Mayan Lab at Mount Sinai, and today I'm going to present gene and drug landing page aggregator. So high throughput methods for screening small molecules and generation transcriptomic data have created an abundance of data related to genes and drugs. And additionally, the ability to store and make this data publicly available in various databases and web applications has become increasingly simple and inexpensive. And as many biomedical resources provide information about variants, genes, proteins, drugs, and small molecules, as dedicating landing page, combining these landing pages in one central location would enable more widespread access to gene and drug knowledge. So CFDE, Gene and Drug Landing Page Aggregator, is designed to accomplish the same by gathering all primary resources and databases that provide gene and drug landing pages into one site with a simple search tool that only requires the user to enter either a gene or a drug name. The landing page provides a search bar for querying gene symbols and drug names of interest. A drop-down on the left from the search field allows to switch between the gene and the drug search types. Search results can be additionally filtered by whether a resource is NIH Common Fund supported, and by the source type, whether a resource is a primary source of data generation or aggregated the data from other resources. After submitting queries, the website displays a dashboard of resources presented as cards. The results are filtered by availability of the hyperlink and sorted based on whether it is tagged as a primary source and the number of clicks on the card. The structure of resource cards for genes and drugs is identical. The header of the card displays the resource center affiliation, the website logo, and the CFD affiliation. The body of the card displays a screenshot of a landing page of the corresponding resource, a short resource description obtained from the resource page, and a hyperlink to the resource. Finally, the footer of the card displays a hyperlink to a page of a gene or a drug on the requested resource. The gene and drug resource pages show an overview of all gene and drug resources from which gene and drug page aggregator collects the data. And every resource card provides the same information as a results card, center affiliation and CFDE affiliation, a logo and a screenshot of a landing page, a short description, and a link to the resource. And cards are sorted based on the primary source and the number of times they've been clicked. Gene and drug pages aggregator is a React application that uses Next.js to render pages server-side. It generates indexable and cacheable static pages. We utilize incremental static regenerated page caching to provide the benefits of static web pages, including search engine optimization, and page caching properties, but recompute the pages after a period of time to ensure the information is kept up to date. And the most common approach for aggregating data is to aggregate it into a unified database for querying, but it quickly becomes unsustainable as variety data types of different granularity are integrated. And our approach is different. Our approach is not to store and serve the information, but to query resources on demand and use each resource to serve the information they were designed to serve in a meaningful fashion, starting with primary sources. This catalog manifest lists all of the gene and drug center resources, including their descriptions, categorization tags, callable functions to generate hyperlinks based on a drug or gene query. These manifests are resolved at query time based on a gene and drug information provided and filtered based on the availability of hyperlink and sorted by whether the document is tagged as a primary source, and page rank based on number of clicks. To display the availability of genes and drugs across resources, we generated accessibility matrices. Genes and drugs are listed in columns of these matrices and resources in rows. Bright color represents the presence of a gene or a drug in a resource, while dark colors indicate its absence. The Downloads tab provides access to the manifest of gene and drug resources, as well as matrices that describe specific gene drug availability across all resources. These matrices were generated by querying many gene and drug terms in each resource and will be updated periodically. And I'm going to show a short demo. So to demonstrate the use case, we searched on the gene Klotho, which is known to be downregulated in aging and is important for healthy kidney function. Gene and drug aggregator resolves the gene resources manifest for 43 gene center resources when you click the search, when I click the search button. Okay. It converts the gene names to different notations, such as ensemble ID. For example, GTEx uses ensemble ID, and if you open a GTEx, you can see that internal notation of Klotho in GTEx is in ensemble. Uniport converts to, it can convert to Uniport ID on or any other IDs or notations that are internal to resource, since some resource use gene notation that is different from interest gene names. And after resolving gene IDs, gene and drug aggregator requests gene-centric resource which correspond to resolved gene IDs. And it renders corresponding gene cards containing a hyperlink to the gene page on the resource if the resource responds with an HTTP status code 200, indicating that a request was successfully completed. Here's an example of data you can obtain. You can obtain expression level in different tissues from GTEx, or glycoprotein data from GlyGen, or geosignatures from SIG-commons, or SNP trace from GWAS, and it's plenty of resources. I'm running out of time, so I'd like to thank members of Maya Lab for helping with this project and developing this project, and NIH for funding this project.